Thanks for the introduction, and thanks, Hui, for the invitation. Thanks for coming to my talk. First of all, I would like to introduce our lab, the Brigham. So this is a map so this probably know that this is Harvard Medical School, the Longwood area. The many hospitals here, Brigham is one of them, children's hospital. And on this side, you have the Funway Park. So this is a Red Sox home field, and there's a tiny building belong to Mass General Brigham. So our building is here. So there are several labs here. So my primary affiliation is a psychiatry neuroimaging lab, and so we have a group of neuroscientists and computer scientists. So we are trying to using different neuroimaging modalities to understand brain abnormality related to psychiatric disorders such as schizophrenia, depression. And we work closely with CF Weston's RMI and also Ron Kingney's surgical planning lab. So they are developer of 3D stressor software. And many of my colleagues also have a joint appointment here at MGH. So so myself also have an office at 10th floor. So maybe we'll see each other pretty often later. For a long time, we have been using diffusion MR as a tool to study structural abnormality related to mental disorders. And this figure probably may be familiar with, you may be familiar with this one. So this is a diffusion stratigraphy. So basically it's a set of fiber bundles derived from the imaging. So just give you the information about the structural connectivity between different brain regions. But for psychiatric disorder, I think the clinicians really are interested about the functional disorder. For that, I think functional MR or other functional imaging modality like EEG, MEG may be useful to provide information about the information flow between the structural pathways, which is not really provided by diffusion MR. So that's motivate our work about combining different modalities to understand brain network. As you know, functional MR is really a massive research field. And I would like you this next figure from this picture to show different direction functional MR, just to place my talk in different to have a good idea about the talk. So you see three axes here. So this is spatial, temporal, and the topological. Basically show the different resolution of the brain network at different scale. So if we try to minimize the temporal resolution, then this classical way of consider brain network is a static network. Basically, the connection strength really doesn't change over time. It's a kind of static connection. And there's many tools have been developed to quantify the connectivity, like functional connectivity, effective connectivity, and also causality, graphical theory, and different many approach. But if we increase the temporal resolution, suppose we have a long scan, I want to analyze the time-dependent connectivity over time, or want to investigate scan-to-scan variability. So that's come to the time-resolved connectivity. And people have developed different measures to quantify the variability over scan as a feature of studying brain networks. But from neuroscience view, it's really, I think we need to better understand the spatial temporal dynamics to understand how brain works. And that may be useful to understand the basis of mechanism psychiatric disorder. So basically, that's really motivates our work. So this talk really covers two subtopics about static connectivity and the spatial temporal dynamics. And this, I think the basic approach is really about linear regression. I think everyone knows linear regression too, but we kind of generalize linear regression in different dimensions, different approach to get information in different dimensions. So the static connectivity, we work on the approach called the causality analysis, Granger causality analysis, if you heard this term. But our contribution is a new approach to go to the frequency domain Granger causality to understand specific information flow in different frequency band. Spatial temporal dynamic is about you use a stochastic optimal control to understand why there's a spatial temporal oscillation in brain networks. Yes, that is the first one. In functional MR, we have a time series data from different voxels of different brain regions. Functional connectivity is a standard approach to quantify the interaction between different regions. So it's exactly the correlation coefficient between two time series data. It's been used in many applications. For example, you can select one seed region and compute the FC map of all voxels connect to that region. So that gives you FC map. You can, so that it has been used in, for example, in TMS to localize brain target based on FC map. So another application is a connectivity matrix. It's giving information the global network, functional network connection, basically 25 the interaction between one region and another region here, and also can be developed to define brain atlas. But it has limitation because it doesn't provide information about the direction of the interaction. For that, people develop different approaches to compute the effective connectivity to understand the direction of information flow. And for example, this diagram shows that two regions A and C may have information from B. You may see A and C are correlated, but actually they are not really, there's no direct information flow between them. So understanding the direction information flow can recover the structure of the network. And in effective connectivity matrix, very similar to this one, but you have an asymmetric matrix. This is the connection between two regions or two voxels is asymmetric in this sense. So, but different from FC, there's no unique definition in effective connectivity. There are different approach. So I'm not going to not able to go into all the literatures, but I'm going to talk about one specific approach called based on Granger causality analysis. Granger causality is really based on the model for the time series data. It's called autoregressive model. It's probably one of the basic approach for modeling time series data. Suppose we have a vector variables. So each entry represents the signal from one brain region. And we have a lot of brain regions here. So we pack them as in a vector, and T is the index of time. So autoregressive model or vector autoregressive model is just, we can predict the current measurement using past data. And there's some prediction error here represented by U. So this model can be written in a compact form based on using some time lag transform, or Z transform. Basically, Z applied to as T is kind of shift in the time space. We can pack everything in the function is Z like this form. So this exactly the same as this one. We are reaching a compact form. The advantage of this compact form that we can write the power spectral density function based on this function form. So for example, this time series data, the power spectral density function can be written in this way. We can replace Z by e to the power of I theta. Theta is the frequency. So for any fixed frequency theta, so we have a matrix here, Sx is the positive definite matrix, give you the interaction of time series data at different frequency. So we'll use these two in the later analysis. But this is really a fundamental to, but keep in mind that in large network analysis, we have to make sure the network is stable. Basically, all the eigenvalues of the system matrix has to be stay in the unit circle in the complex domain to make sure the model parameter is physically meaningful. And for Granger causality measure is really a generalization of the R-squared measure from statistics. R-squared measure probably learned that from the linear algebra 101 or statistics 101. So it's really a way to quantify the linear dependence of two variables. Suppose we want to explain x1 using x2. The linear dependence R-squared measure from x2 to x1 is 1 minus the residual variance divided by total variance. Or we can write in this way. So 1 divided by 1 minus R-squared total variance divided by residual variance. To be more specific, so let's suppose we have two variables. This is their zero mean, and this is their covariance matrix. The linear dependence measure here is the total variance before regression and total variance after regression. So this is exactly the same as the variance of x1 divided by the variance of another variable we call x2.1. x2.1 is x1 minus a variable, another variable is f times x2. f is a coefficient. Basically, we want to regress out the influence of x2 from x1 to minimize the variance. So I hope you're not really bored about this definition. The key point that we can remember the logic that we defined R square measure by introducing a new variable that has the minimum variance using some kind of filter here. And similar thing, following similar rationale, we can define the Granger causality measure, or if Granger causality measure, because the formal definition is from this paper. Suppose we have two time series data x1 and x2. So we want to estimate the influence of past signal x2 to counter value x1. So definition, the exponential Granger causality measure is this expression. So let me go through each of terms. So this term is the expected value of x1t given its past data. Because it's randomness of time process, so this expected value is not exactly the same as its true value. So there's prediction error here. And similar here we just, if we add the past value of xt to predict current value of x1t, so we are expecting to have better prediction accuracy. So the variance in the denominator is supposed to be smaller than this term. So the difference quantify how important the past value of x2t to predict current value of x1t. So that, but another compact form like this. So following Gaussian assumption, if everything is Gaussian for Gaussian process, this complex expression can be understand like entropy rate of x1t divided by entropy rate of another process. So it's kind of similar to the definition of R square. It's kind of ratio of two variance. But x1t is a random process that is defined by x1t minus some function of x2t. It's very similar to the static filter that we see before, but here it's a really causal filter, minimize entropy rate of x1 tilde t. So now if we put it this way, simply everything seems to be trivial, but actually this is not really the way that Granger or Greg define their causality measure. And you may all have heard about the Wiener filter if you are familiar with signal processing theory. But actually this is different from the Wiener filter because in the classical signal processing theory, we are trying to minimize the mean square residual, not instead of entropy residual. So that is the difference between the classical prediction theory. And there's some different generalization causality measure by introducing instantaneous causal and non-causal filter. Basically, one by change the time range of the x2 variable. For example, we can use the instantaneous causal measure is similar formulation as before, but the new variable is we add the current value of x2t into the prediction. Then we have supposed to have a lower entropy rate. So the difference between the instantaneous causal and the strict causal is that we can have understanding about how importance of the current value of between in the prediction. Okay. So any questions so far? Okay, so if so following similar rationale, we can introduce a non-causal filter, non-causal Granger causality measure, because by introducing a filter that uses both past and the future to minimize the entropy rate. So even though they look complex, but they can be computed based on the unique vector autoregressive model for the entire process. So I'm not going to show the expression how to compute that, but give some results. You have to see the instantaneous causal and non-causal. Hope that will be useful to understand the difference between them. So I think one of the but following the rationale, so we define the filter to regress or the influence between network can define the frequency domain causality measure. But actually, frequency domain causality measure was already defined in the original paper. Let's see, but it's different from the regression-based definition. So this is the power special density function that we can see in the first slide by expanding different terms. So we can take the inverse of this matrix that has B matrix. So definition of frequency domain causality measure, we have a function, another causality function is a function of frequency. It's the difference between the ratio between the original determinant or original time series divided by a new power special density function. This really have a complex expression, so I'm not too able to show the insight that they define this matrix. Basically, they're trying to decompose the residual process into D2 process, and so as Greg said in his paper, he said this is an empirical solution, trying to make sure that the average frequency domain Granger causality matter equal to the static Granger causality matter. So that's one of his motivation. The physical meaning here is really not clear. Basically, it's based on the decomposition of this residual process, not really defined by some filter. Given that we have a filter, a new process based on X tilde, so in our paper, we introduce a new Granger causality matter, the ratio between the original process divided by the power spectral density function of new process. New process we know is really based on the data, based on the filter, and also it has very neat, very simple expression as before, and also surprisingly, probably not so surprisingly, is also satisfy the equation that Greg was looking for. This is average in the frequency domain Granger causality equal to the static Granger causality matter. So let's put them together in the one slide. So basically, this is the original solution, a very similar expression as this one. Only I think the difference is the factor of the filter expression. So this one is more complex, but that G is not equal to A11 inverse, so that's why they're different. So let's see how they're different in the real case. But in the real case, so we are not really care about the pairwise Granger causality. We are looking for a large network. So brain usually have you can separate brain in hundreds of regions. How do we quantify the information flow between two nodes in a large network? So this is, in that case, we have a really high dimension time series data, X1, X2, and also other nodes and time here. So in this case, the Granger causality matter already defined by pairwise definition. But the pairwise Granger causality matter really doesn't make sense here because there's indirect pathway to transfer information between them. So, but following the idea about regression, so we can define conditional GCM in a two-step process. For example, we can combine everything, all the variable in the new variable X1, 2 complementary set of 1, 2. We can first regress out this variable to the two-dimensional process by using the entropy rate minimization process. Then we have new two-dimensional process. Then here, for the new process, we apply the standard Granger causality measure by regressing out this new variable from this variable here. So in the end, we define the conditional Granger causality measure like the difference between the original power spectral of this process divided by the power spectral of this process. So the difference really provide information about the additional information of X2 to predict the future variable of X1 given other variables. So this kind of Granger causality measure, even though it's two-step process, you can get the solution based on the unique, a large one-dimensional, it's a unique vector autoregressive model without using the two-step process. Basically, just look at one closed form solution for two-step procedure. Let's see the performance difference between them. So this is synthetic data trying to compare the difference between the standard Granger causality measure. We just show the blue line here. Red line is the strictly causal. Basically, we don't use the current value of X2 to predict the current value of X1, and also this, and also a non-causal filter. Basically, if we use everything, ignore the direction of time to see what's the difference between them. And you see the two diagram here. There's three-dimensional time series. So the direction really shows the direction of the A matrix, the entry of A matrix. The parameter just these are the parameter of the autoregressive model. So as a first order autoregressive model. Also, the only difference is that this one has direction from 3 to 1. This one does not have the direction from 3 to 1. Let's see the difference. So let's see, this one is different from this one. So this shows the conditional causality from 1, 2, 3 given x2. So see this one, because there's no connection here, so everything is 0 here. But the non-causal here is not 0 because it totally makes sense because the future of x1 provides information about x3. But the straight causal and the instantaneous causal all make sense here, and similar information we can find here. But another interesting result that this proposed method is the red line is very different from the blue line in some curve here, especially in the low frequency range. The model parameter here really configure about the low pass filter. Basically, all the information should have low frequency range, but the original definition really looks arbitrary. Really does not really provide insight to understand the interaction in the low frequency range. Let's see some in vivo data here. We did about the same thing for the 3-net brain network based on the Thomas Yale's brain parcellation using the Human Kinetome project. We separate, we use the standard Granger causal measure, our proposed straight causal Granger measure, and instantaneous causal and non-causal measure here. So I think there's, so first of all, we see that the instantaneous causal and non-causal very different from the straight causal measure. So, and they are similar to each other. So this just means that the time series data may have a very strong spatially coupled white noise. So this maybe because that the signal to noise ratio may be low, so that's the signal, the measurement itself, there's a lot of measured spatial coupled white noise in that. So basically, the current value has the strongly predict by measure value to predict another time series data at the same time point. I think maybe it can be solved by increasing the sampling rate. And another difference that the proposed measure is straight causal measure is different from the standard causal measure. So basically, the red curve is very different from blue curve here, and the red curve has, I think, very interesting shape as the 0 to 2.1 hertz frequency range. That is the range that we are interested in functional MR. So that's the range of hemodynamic response. See that, for example, the dorsal attention to ventral attention. So there's non-zero measure here, but the other way, ventral attention to dorsal attention is almost 0. Maybe the interaction between these two regions is only one directional. That may be interesting to test from other approach. So that's basically the technical detail about the function frequency domain Granger causal measure. But how do we use that in practice for schizophrenia research or depression research? So we have been developing a pipeline trying to combine structural connectivity with functional connectivity to study and the information flow in different networks. And following this conditional Granger causal measure, we can define the pairwise effective connectivity measure between each pair network measures. We apply this approach for public data set that have both control schizophrenia and bipolar ADHD. And following this kind of definition, so we project the network effective connectivity measure of different patient profile to different principal component space. Interesting, we can find that the 3-type disease can really be separated very well in the imaging feature domain. So schizophrenia are very different from bipolar patient, even though they have very similar genes. And bipolar ADHD, they have some overlap, kind of consistent to the clinical, observation. They have pretty large overlap in clinical study, but they can still be separated in some sense. They have, each of disease have different set of networks. Some of them are consistent with some early literature. So we are trying to continue developing this pipeline so that to make it useful in other studies. Okay, I think this more or less summarizes the first topic, basically the frequency domain Granger causality measure. So trying to shift to the second topic. So this effective connectivity and functional connectivity are really based on assumption that the connectivity network doesn't change over time. Is this really true? For example, this is the figure from our paper. So we have to the same seed region compute the FC map from different scans of the HCP data. See, even though the global features can similar, some region still always positive, and some large regions remain negative. But if you go to the fine details, you see that quite large variability over scan. And another paper also showed the scan-to-scan variability of the FC matrix. Trying to see how whether it can be reproduced from scan to scan. So, for example, if you scan the same person from 3 scans, this is the similarity measure between the FC matrix. I think in some sense it could be, the similarity could be very low, but it really depends subjects and also the scan interval. So for some subjects, more or less similar, but some subjects, the similarity really decays significantly by increasing intersession interval. So there are many reasons that we people have been just trying to explain why it happened. So one reason that brain is really changing over different states, and we so that's why I can see the variability. And also we really need long scan time to make sure to increase the interscan variability. And also it's always helps to increase the signal to noise ratio. And also the within scan variability also has been observed. For example, this figure from this paper showing that if we apply different window to the time series data, so you see the FC map also change very rapidly over different windows. So for example, this time window gives this FC matrix, this gives this one. See, some region is very positive here can become very negative from another time interval. Basically, it's really quite large oscillation. For those you haven't really seen about the functional model data, so this video I found from Wikipedia showed the fluctuation in FC functional model data. So basically, you see the oscillation at standing wave, basically oscillation at the same place, and also the traveling wave, the energy spread over other regions. And does this result really show that the stationary assumption or the static assumption is wrong? Actually, it's not. I see a paper from Thomas Yale's group show that really a stationary process can have strong oscillation or fluctuation in the short time range. And also, even though it's fluctuation in short range, so if the scan is really long, it also makes sense to use FC functional correlation matrix to define two-sided brain network. But from a neuroscience way, it's really interesting to understand the spatial temporal dynamics to understand how brain works. So now we have evidence show that brain really oscillates at short range. So so every time point is different from the other time point, but from long time range view, it's kind of static. So how do we understand brain network? So I found this picture maybe helpful for understand brain networks. The solar system. So we know that the planets are running around the sun in the really long time scale. Really everything, every planet have a kind of fixed orbit with fixed period, but the relative location of different planets is different from the previous time point. So basically, understanding the trajectory of this the dynamics is really useful to understand the underlying law that's driving the functional dynamics. So this really drives the kind of preliminary work trying to define brain trajectory. So what is the trajectory of brain network? So I don't have a clear answer for that, but I try to define the approach following the standard engineering heuristic. Basically, we'll use the kind of geodesic regression or basic generalization of linear regression to find what is a model that can explain the dynamics. So geodesic, so if it's basically a generalization of straight line in the Euclidean space. If I'm studying on the endpoint, you know the shortest path is a straight line. geodesic is generalization of straight line in the curved space. For example, this shows the geodesic between a place in China and the Boston. It really is a great arc between the 2 points. So suppose we have some observation with time varying FC functional kinetic matrix. So if we are able to find a continuous model that that really fits the data very well, and their model really driven by a simple principle on shorter path or minimum energy, maybe that model is useful to understand brain dynamics. So that's really the heuristic and for this for our work. So this become a little more complex, but we may go through this part. So when we define the shortest line or straight line in the Euclidean space, we have a starting point and end point. Then we have a way to measure the path length. The shortest length gives the straight line. Similar thing can be generalized in the curved space. So we have a starting point. So that's the FC matrix that we have observed at time point t, and another time point FC matrix and time point 1. We are looking for a smooth path connecting these 2 endpoints and by minimizing some energy. But given that we're assuming that the FC matrix is only an observation of dynamic system, so the dynamic system really is the model for modern brain dynamics. X is the vector of functional MR data that we have seen. So it's driven by a dynamic system AT here, and from XT, if we're able to sample XT very quickly at one time point, we have a observation of the FC matrix. This comes the sliding window FC matrix. So we know that if this model is driven by this dynamic system, that P of T has to satisfy this dynamic equation. So they are kind of model consistent to each other. So we are looking for a path connecting end and point by minimizing certain path lengths and energy. The solution to this problem gives you a model, parametric model, so that you can apply to feed the data. Basically, in this Euclidean space, you have a straight line here. So X is the functional MR data, and P is the observation, the FC matrix. A is the system dynamics, is unobserved brain dynamics. So what type of solution can we get from here? So I think from physicists and statistician of different insight about what function to be used. For example, I go through there's some standard solution for this. I think the physicists may tell you about, maybe you can look for cause function that minimize the kinetic energy if we think everything to particle. So here x dot t is the kind of velocity field. So if we try to minimize the kinetic energy for transferring the mass forming a Gaussian distribution to another Gaussian distribution, so probably we should use this cause function. So this problem is related to the optimal mass transport. The solution is really well studied, especially in recent few years after Villani got the Fields Medal Prize. So the solution gives you that the P of T is a quadratic function of time, and the A of the underlying optimal system dynamic is a symmetric matrix, even though it's time varying over time, but it's symmetric. Basically, it doesn't really provide the asymmetric interaction in brain networks. Maybe a statistician may tell you that in the L2 norm or the kinetic energy doesn't make sense here. We should the cost may be penalized based on the distribution itself. Maybe use a weighted L2 norm here. So from this one, I found that this solution here provides the solution the exact same as the Fisher-Rao metric. So I think some of you may be familiar with this classical solution in some textbook. The P of T is a exponential function of time, and the underlying dynamic is a static function. It's basically a constant function. It doesn't change over time. This is the same as Fisher-Rao metric, even though I think the classical literature Fisher-Rao metric is really not motivated based on this kind of mechanical view. It's from the Kullback-Labrador divergence, a different view. But here, does this really make sense for brain dynamics? I think maybe from biological viewpoint, we are looking maybe interested about something else. So here we are thinking brain is a network. A is a system dynamics, trying to transfer information between different regions. Maybe it costs energy when it's transfer energy between regions. So in that sense, maybe the cost will be some the energy of transfer energy. The cost of should be the energy function that quantify how frequent that transfer energy between the regions. So that should be a function of A here. So I consider that the A may be asymmetric. Maybe the symmetric interaction and asymmetric interaction have different costs and energy. So maybe a function of energy cost. In that case, found that they give really some interesting solution here. So this is a solution given by this cost function. It's really complex, but I think the interesting part is this term. You see the exponential asymmetric function of time. We know that if exponential asymmetric function is a rotation matrix, so if it gives information about the rotation of energy different brain regions. And the analysis system AT matrix also time varying and asymmetric matrix. So I found that's really interesting. It really give insight to understand brain dynamics. So it really, the rotation matrix gives information about spatial temporal oscillations. I think maybe to one step further, I think this network may be too simple to explain brain dynamics. It's really not autonomous function. If we add makes everything more complex, we can add some external input to it, very similar to the autoregressive model. If we add external stimulation or spontaneous activation, different network, we can also make the model more complex by adding the input to the system. So the corresponding FC dynamic should be consistent to this one. And there's also general, very interesting theory behind different equations, and we have some closed form solution. But the key point that this is generalization of the previous solution from the previous slide. So the minimum transfer energy function also generates oscillation in the network. And see how well this performs. So now we have several approaches to define a continuous path in the complex domain, in the curved space. And similar to the Riemannian geometry, so when we define the curve, we define the starting point and also a tangent direction. So then the curve just run it by itself, give a curve. So if we have a different sample matrix based on the starting window of time series data, so P1, so this is our sample, and we can fit the noisy measurement, so this here, by a continuous path that's really parametered by few model parameters. So maybe we can try to see which of the model that fits the data well, or maybe that's useful to understand brain dynamics. So this shows us only one slide showing the fitting results. So this is measurement that we have seen from previous slide. So at each time point, so this black dot here is basically the measurement at one particular entry here. And the blue, green, and red curve really is the result from the mass transport information and theory, and also the energy, transfer energy theory. See that the two curves, the blue and the green curve, really can straight, because one is quadratic, the other one is exponential. But the transfer energy gate function-based one has oscillations, even though they have the same number of parameters to parameter the trajectory. So the fitting result is much better and also kind of interesting. For example, you see the oscillations of this one entry of the network really can be modeled tracked by this curve. And also interestingly, that's why the solution also gives a model for the system dynamics. This is A matrix. As you can see that the overall also change over time. I mean that the different brain also interacts at different time point. So that different pattern interaction. So I think this is preliminary result. So for this network. So hopefully I found this interesting. So this will summarize my talk. So basically we explored two topics. The first one was frequency domain Granger causality measure for static static brain kinetic analysis. I think one key point that we have the entropy rate regression-based filter. We think it's probably more reasonable to study the interactions because its physical meanings can be better understood. So another about the theory trying to model the spatial temporal dynamics. Basically, we found that the spatial temporal oscillation that we found from brain network maybe because that the brain trying to minimize the energy of transfer interaction between different brain regions. So that's only maybe hypothesis generating theory, but needs to be further validated. Okay, before I conclude my talk, I will maybe give a brief overview of our other talks and our other works in our lab. So this talk is about modeling for faster, better imaging for functional MR. Our group, our lab. So I work closely with Yogesh and CF to develop different models for diffusion MR. So I think this, as you know, diffusion MR is the way to study the microstructure brain tissue or brain axon of cells. Standard approach trying to see the crossing fibers and fiber microstructure and to study cartography. And because we are able to acquire image faster and better in the new scanning protocol made driven by a colleague here. So now we are trying to, similar to the way in functional MR, we are trying to explore different generalization, different MR to different dimension. We explore different temporal dynamics and also and the coupling diffusion with other imaging modalities. So if interested, we can talk about more detail about here. Maybe invite our colleague here to give a different talk. So that's all my talk. Thank you.